In this video, we will discuss about the isolated cluster T cells has enhanced cytotoxicity. So far, we have understood that the isolated uh, T cells present either in the singlet form or in the clustered form, and they are usually clustered with the tumor cells or with the APCs. And from the bioinformatics analysis that we have discussed in a couple of our previous videos, we have seen that the clustered T cells with APC and also with the tumors has uh, gene expressions changed in a way that will favor its enhanced cytotoxicity. In order to validate that observations, what they did is they took the melanoma cancer cell patients and segregated the T cells and the tumor cells in different populations. That is tumor cells alone, T cells alone, that is T cell singlets, and the T cells with the tumor cells clustered, the T cells with the APC clustered, using the fluorescence activated cell sorting or the FACS. Thereafter, they used REP, that is rapid expansion protocols, to expand these populations, and then did the in vivo studies and also the ex vivo studies. So we will discuss in this video the results of the ex vivo studies. In the ex vivo studies, what they did is they took the T cells and co-culture from the different populations and co-culture them with the tumor cells individually, and they check the results after 4 hours of co-culture with the tumor cells of the different populations of the T cells. And first they check the cytokines that are released by the T cells, and in terms of the cytokines, more specifically, they check the INF gamma and TNF, and they are able to see that the T cells from the singlet has very low activations or very low secretion of those cytokines, whereas the T cells from the clustered tumors has very high activations of these cytokines, both IFN gamma and TNF, and the similar high expressions are seen from T cells that are isolated from APC clusters. And these things they have quantified, in the panel C, where we can see that compared to the singlets, the expression levels of the T cells are high for this TNF and also for this IFN gamma for the different patients. Almost all the patients shows a high expression of this IFN gamma and TNF alpha. In the case where T cells are found either from the tumors or from the APC. Thereafter, they also did the ex vivo cell killing assays, or they checked the ex vivo cell killing capacities of these different populations of the T cells. What they are able to see that they checked it for the 11 different melanoma cancer tissues that they obtained from 11 different patients, and they are able to see that in the case of this clustered tumor cells, 9 out of 11 cases they are able to see high expressions of the cell. They are able to see high tumor cell killing compared to the singlet cells in the 9 out of 11 cases. Yeah, and for the case, and in the case where they used or when they checked it for the T cells that are isolated from the APC clusters, they are able to see 11 out of 11 shows high expressions of the tumor cell killings, and that is more than 9 times high expressions compared to the singlet. So this in all together indicates that the cluster T cells gene variations that we have observed in the results that we are discussing from the previous main figure of the same paper, it seems to be correct, and the cluster T cells either from the tumors or from the APCs has high cytotoxicities against tumors or high anti-tumor efficacies against the tumors. In the next video, we will discuss the other remaining part of this main figure. It might be that might reflect the results for the in vivo studies. So far the ex vivo studies results are in line with the observations that they have seen in the results that are presented in the previous main figure. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.